Do you see my slides correctly? No.
I see the view that you see on your desktop when you're working on them with the ribbon on the side. Now
Okay.
I see it correctly.
Awesome. Okay, do you want to go ahead and get started?
Great, yeah, take it away, Amelia. Thanks everyone for coming.
Yeah, awesome, everybody. Thank you for coming, especially our panelists. So welcome to the first current topics of the new year. So this is a seminar as part of a series that we organize with the Epi-Ideas group, Communication of Scientific Data sub-working group. And today's seminar is co-sponsored by Epi-Ideas and the Departmental Curriculum Committee. So, sorry, hopefully you've heard of us, but Epi-Ideas is a working group of students, staff, faculty, fellows, and yes, students, fellows, staff, and faculty who work to promote inclusion, diversity, equity, and anti-racism in the department curricular and all co-curricular activities. So we host workshops and trainings on communication of epidemiology to broader audiences, and then work to promote public health advocacy in epidemiology. So for the past few years, we have hosted 2 seminars a year on topics ranging from the role of epidemiology in shaping public policy and evaluating impact of policy on vulnerable populations, to methods of disseminating academic research, including via Twitter and via mass media. Our last seminar in the fall focused on ways to engage with the local Baltimore community in our research, and today's seminar will be focused on the use of race. So these seminars were prompted in part by the events of the summer and the department's statement on racism as a public health problem. As I'm sure you are aware, as a department, we made several commitments towards changing our research and educational practices, and this seminar is one step towards honoring those commitments. So this is not all we're doing. However, you should have received this email from Epi-Ideas and the departmental leadership earlier this week that outlines some of the steps we have taken to evaluate needs of our students, fellows, staff, and faculty, and our strategic plan for the department moving forward. This is still a work in progress though, and I know that Dr. Brawley and the department leadership would appreciate any feedback you have on this. And also in terms of curriculum, several changes have been made to the introductory methods course, and Dr. David Towdy will talk to you briefly about the work they are doing.
Thanks, Amelia. And just very briefly, I don't want to take more time than necessary, but just to say from the perspective of the Departmental Curriculum Committee, this has been identified, both how we deal with race and racism, and also how we deal with other societal constructs, whether they be gender or socioeconomic status, et cetera, has been identified as a priority by the Departmental Curriculum Committee. Anyone who's in attendance here who has thoughts about how we should be moving forward with this, if there's anything we can be doing at the departmental level to integrate these concepts more constructively into the EPI curriculum, I think we'd be very happy to hear your thoughts. So please don't hesitate to either put them in the chat or contact me directly. And just also to say thanks on behalf of the curriculum committee to both Epi-Ideas and to our presenters today. I think this is going to be a great presentation, and very excited to listen in and learn myself.
Awesome, thank you, David. So in addition to the seminar today, we also have a number of resources that we've started compiling, both academic papers and public media. So we will have Laura post this to the Course Plus site and distribute it elsewhere if you would like. And so finally, all of our panelists are very active on Twitter and have wonderful, post wonderful things. So we encourage you to follow them. We will post their handles into the chat and also the department. And we ask that if you post about the seminar today, to please use the hashtag Epi-Ideas. So with that, I'll pass it on to you, Catherine, to introduce our first panelist.
Okay, well, fantastic. Thank you, Amelia and Dr. Dowdy, for kicking us off. I believe Dr. Dean is going to pull up her slides while I'm speaking to save us a little bit of time. So with that, it is my pleasure to introduce our first speaker, Dr. Lorraine Dean. Dr. Dean probably needs no introduction, so perhaps we can think of this as an opportunity to recognize her many contributions to our department and her leadership, which has in many ways helped bring us together today. So Dr. Dean joined the Department of Epidemiology in 2016 and was recently promoted to associate professor. She is a social epidemiologist by training, and her work focuses on the social determinants of cancer survivorship. She also conducts research examining factors influencing HIV pre-exposure prophylaxis, initiation, and retention. Dr. Dean is highly engaged in teaching and mentoring. She teaches advanced social epidemiology and co-developed a service learning course, Methods for Assessing Power, Privilege, and Public Health in the United States. She has received the Golden Apple Award for Excellence in Teaching and the Delta Omega Award for Innovative Public Health Curriculum. Additionally, as you may know, Dr. Dean has been involved with Epi Ideas since its inception in 2016-2017. And while she would probably want me to emphasize the flat organizational structure of our work group, I think it's fair to say she has had a critical role in advancing our efforts within the department. And she's someone that other departments and other institutions look to for advice when considering their own initiatives. So thank you, Dr. Dean, for all that you do for our work group. And on behalf of the planning committee for this event, thank you so much for being with us today. So without any further delay, it is my honor to welcome Dr. Dean to the virtual podium to discuss the use of race as a variable in social epidemiology. Dr. Dean.
Great. Thanks so much, Dr. Foti, for your wonderful introduction. Also like to acknowledge the work of Amelia Wallace, Dr. David Dowdy, Laura Camarada, and the many other people who worked, and my other panelists who worked together to pull this presentation together. So each of us as the panel are going to give you some quick presentations. I'm going to speak for about 15 to 20 minutes that will give you an overview on the use of race in social epi that is going to leave you wholly unsatisfied. I promise you that. But at least it'll give you some initial ideas and thoughts about how we think about race, the background on race, our practices for measuring race. And then at the end, I'm going to talk about when you talk about race as you're writing your papers, what are some things that you might want to consider and do? So part of the question of the hour, right, is what is race? From a social epi perspective, those conceptions have changed over time. Technically, the word race initially referred to a part of the taxonomy. So that's that genus, kingdom, phylum, right, all the way down of human taxonomy. That underneath of subspecies, there was something called race. And the idea was to classify people based on these phenotypic differences, but we realize now that those phenotypes don't match well to our underlying genetics, and they don't match well even to our current race categories. So I have not seen that in any of the recent taxonomy hierarchies anytime recently. I'd also say that what we think about in terms of race is often conflated with genetic ancestry, which I think Jan Wojcik might talk about later. But genetic ancestry is about exposures and selection that happen over time based on people who live in similar geographies, have a shared language, but that is not the race that we are often talking about and measuring when it comes to studies, especially in the United States. However, that gives you some framing for why for many years people talk, in even medical science, people talk about race as if it's biological. And in fact, in terms of the US medical curriculum, the idea of race was brought in about 1790 by someone named Benjamin Rush, who asserted that, particularly at the time looking at the enslaved population in the United States, who was Black, asserted that Blackness was actually a form of leprosy. And there were others that tried to talk about slavery and used essentially a racial supremacy ideology to talk about why Black people were the ones who were enslaved. But even from early on back then, people were rebutting this and refuting this. And one of those people was Dr. James McCune Smith, who was the first Black American to earn a medical degree, to publish in medical journals, and essentially started using medical science to debunk this idea that these race differences that they were seeing were due to biology. So then, not due to biology, so then how do we think about race? As Dr. James McCune Smith would have said, even back in the 1800s, and we've still been fighting this battle for over 100 years, right? Race is a social construct. Even anthropologists have agreed that race is a social construct, and it means that there's greater variation because there's greater genetic variation within racial groups than between them. And even in terms of medicine, if we look at, as an illustration, if we look at something like high blood pressure, we know that in the US, there are particularly disparities in high blood pressure and Black-white disparities that contribute to differential blood pressure prevalence and mortality. So if you look at this graph where we see a comparison of blood pressure rates across the world, if race in the way that we think about it was truly something biological, then we would expect that the US born Blacks to be similar to other Black people who were displaced from Africa, were stolen from Africa, but might live in other countries, or that they were very similar to potential countries and countries from which people might share their genetic origins. But we actually don't see that in our studies of US-based Blacks. We actually see that the hypertension rates are very, very different for Blacks coming from very different places. So then what actually are we measuring when we talk about race? Well, essentially we're measuring, as Nancy Krieger would say, racial categories reflect social and ideological conventions, not meaningful natural distinctions. That is to say that race is measuring something that's socio-political. And essentially that this racial worldview that we use is essentially used to assign benefits to some people while denying benefits to others. So these present day inequities that we see between racial groups is not something about biological inheritance, but instead is a part and due to social and economic and educational political circumstances that are differently levied upon people of different races. And if we use even as one example of how race is thought of in different ways, it has and can change over time, and is really a result of our socio-political orientations, take someone like President Obama, who we call our first Black president. He was called our first Black president. He had a white mother and a Black father. But because of our history in the United States of using something called the one drop rule, it was this racial supremacy belief that having one drop of blood that was from anyone else's race that was not white, that is this kind of tainting the purity of white bloodlines, right? We needed to designate that in our society. So this idea behind the one drop rule meant that if you had any parent who was not white, then you would be classified by the race of that other parent, the non-white parent. And that's how it worked on birth records for a really long time. So when President Obama was born, his birth record would be classified by the race of his non-white parent, his father, and he was classified as a Black president. He wasn't classified as a president when he was a baby, but he was classified as a Black man. However, since 1989, the National Center for Health Statistics has actually changed how they've tabulated birth data. And now it's according to the mother's race. So technically when he was born, he would have been listed as a Black child, but if he had been born 10, 15 years later, he would have been classified as a white child by the National Center for Health Statistics. Yet we still refer to him as our first Black president. So that was just one example. There's several examples that we can use to talk about and see how racial categories have changed over time. But essentially, race as a social construct means that these race-associated differences that we see are changeable because they're due to something that's not inherent necessarily to our biology. That even a person's race might not be stable over time. And in fact, there are several studies that show a race discordance between what people are classified at birth and what they're classified as at death. But then also that race is actually a crude proxy for these other social factors that we might not be measuring or explaining. They could be things like racism and discrimination, but they could also be embodied expressions of those things like racism and discrimination. And they could also represent other social things that we don't measure well or have other proxies for. So in terms of thinking about how you go about measuring and reporting race, one question you might be up against is, what racial categories should you actually measure? So the first thing is to think about, again, if you're considered as race from a social construct, means that you need to have a conceptual framework that actually explains why you think race is related to whatever outcome that you're considering. So base it on your conceptual framework, base it on your analysis plan or the sample that you have. If you're working with a sample that you want to compare to a broader population, you could consider using the census-based measures for race or the census-based categories for race, but that has changed substantially over time. And even if you look at the 2020 census asked people who classify as white to further identify as German, Irish, English, Italian, Lebanese, and these other categories. but yet has not disaggregated other groups that have asked for disaggregation, especially people who self-identify as in the Middle Eastern or North African category, who often have to check something else that they don't really identify with. So you can use consensus-based measures, but they're not perfect. Junia Howell at Pittsburgh had suggested, in terms of understanding social inequality, that we did some work to suggest that what we should use are the five essential monoracial or ethnic categories. So she did some work comparing a couple of things, the 5-category race, a 3-category race, skin tone measures, and several other things. And she identified that the best way to capture social inequality would be to measure the categories of Asian, Black, Hispanic. Hispanic is an ethnicity, but we can talk about that later. Native American and white. I'll also note that in social epi, we don't use Caucasian, we use white. Nancy Krieger has some work to talk about how the term Caucasian itself is even a racial supremacy term. So we use white, not Caucasian. There are also different types of race that you can measure. You can measure how someone identifies themselves. You can measure how other people identify their race. That we use actually this in medicine and health a lot when we do interview administered surveys, or sometimes the intake person at a clinic might be classifying your race. That's a socially assigned race. And a new measure has arisen called your street race. The idea that what would people classify you as when you were just walking down the street, just based on what you look like. So for example, myself, that identified race is Black, but for people who don't see me and hear my name, Lori, not a super common name for a Black woman, or I don't think people associate it as a common name for a Black woman. I've been classified as white for people who have never seen me before. I've seen it on forms. It's pretty amazing to me. But then there's my street race. I grew up in an area that had a large Latinx population, and so a lot of people thought, because of the area that I lived in, that I might identify as Hispanic Latinx and might even be Dominican. So I actually had 3 different types of races depending on what measurement people would use. Another big question that we get in terms of race measurement is, should you adjust for race in your regressions? This is, again, you're going to leave wholly unsatisfied if you expect to get that answer in the next couple of minutes. But a long story short, social epi largely discourages adjusting for race, because by definition, social epi feels that those social factors are not background information. They're the driving exposures. But let's be honest, I've done it, you've done it. Let's just try to move forward now with what we know now, even if you've done it before in the past. Galea and Link explain it as, what does it mean to enter a parallel universe where everything is the same except for someone's race? That race is a special type of exposure in the way that other epi exposures are not. It's sticky. It's something that affects us potentially even before we're born, and then affects us at all different parts of life. We can't just extract it in the way we might other epi exposures. Race may or may not be considered modifiable. There's a big debate about that that's still evolving. AJE has had some papers where people are going back and forth on that. And then others like Tom Levis have some papers that just say it doesn't even make sense to interpret race as causal, especially if we're going to adjust for it in a regression analysis. That essentially the regression approach just gives us adjusted means for people in different racial groups. And that if we really want to understand what's happening and really make an impact on especially things like racial disparities, that what we should do is stratify our results by race, use effect measure modification. Building on that, Ward and Keyes have a paper that says, well, even if you, for example, have an interaction term to assess effect measure modification for race, even a non-significant interaction term might still mask a disparity. So they examine, in addition to thinking about interaction, look at the group specific differences in the outcome and exposure, as well as effect sizes. All right, to round this out, I just want to give you some examples, some quick examples about things, challenges in manuscripts, or things to consider as you report about race or talk about race in your manuscripts. So the first I'll give you is a cautionary tale. This is a JAMA paper that was about the association of sex and race on weight loss on physical function. The first line of the introduction said, consideration of relevant biological variables. such as sex and race in clinical research. Now, I tell you, I died inside just a little bit when I read this, because I just screamed when I said, how are they saying that race is a biological variable? How could they say that? So I went deeper into the paper. I said, you know what, maybe they're measuring genetic ancestry, maybe they're measuring something else. So I went and looked into the methods of the paper, and that wasn't true. In fact, what they had said was that in this case, the race was defined by the investigator, the research investigator. So it wasn't even a self-reported race or anything like that. Definitely no genetic ancestry measures or anything of that sort. And so I wrote a letter back. I wrote back a comment onto their page, and I said, I'm just surprised and disappointed that essentially at this day and age that an article in JAMA would continue to endorse the antiquated belief of race as biology. And what was really neat, and the reason why I showed this example, is they actually went back and published a correction. They said, you're right, as we think about the way in which we measured this is really not race, any sort of biological version of race that we're trying to measure, and the article has been corrected. So that was for the introduction section. So don't make the fatal flaw up front when you're writing your introduction. But how about your methods and results? Well, JAMA had some other a while ago recommendations for that. One, if you're going to talk about race and ethnicity, tell people the reason it was assessed, justify the use of it and why it was used, and then provide an explanation in the methods about who classified people, the framework that was used, whether the options were investigator defined or self-reported, and importantly, what were you actually trying to measure that you felt like race captured? Even just things like reporting the race and ethnicity of the study population in your results can be helpful. And Kala Visvanathan and I wrote a commentary on this in the cancer world, but we found that except for studies that were explicitly about disparities, fewer than 5% reported any sort of socio-demographic information. So so think about just even reporting the race and ethnicity in your study population results. And then to move into your discussion, just think about the framing that you've used. Don't endorse racialized biology. In fact, Rhea Boyd wrote a paper that told the story of another paper in Health Affairs that talked about these differences in hospitalization for white and Black patients, and said there are these several possible explanations for these disparities, and then went right into one of them is that there are unmeasured genetic or biological factors that increase the severity of the illness for African Americans, right? The problem here is that they essentially ignored race as a social factor, ignored racism as the potential mechanism by which these racial categorizations had biological consequences. But they did later talk about societal factors that have contributed to the disparity: unconscious provider bias, distrust, financial stress, but they still never actually mentioned racism and discrimination. So don't endorse racialized biology. And then finally, in your discussions, if you're reporting on an intervention, as you think about the intervention results by race, if you see differential results by race, instead of framing it to be about these differences by race, reframe it to be neutral to the race of the people in the groups. And what I mean by that is, instead of saying that Blacks or whites did better or worse in the intervention, say the intervention was as equally or not as equally effective. Really, so think about the fact that it's really a product of the intervention if it didn't work well in some groups versus others, not necessarily of the demographic of the people in the intervention. It's something that didn't work for them. So some quick takeaways: in summary, measure what you want to measure. If you want to measure, try to measure things like discrimination or skin color, if that's really what you're interested in terms of race, or genetic ancestry could be another one. In addition to some self-reported race categories, I'm not saying don't measure race ethnicity, it could still tell us something. Consider which modeling approach is appropriate. Like I said, we discourage adjusting for race. That doesn't mean you can never do it, but just be thoughtful about whether or not that makes sense for your study question. Avoid pointing to genetic differences if you actually haven't measured race from any sort of genetic perspective, and then clearly state the factors for which you believe race is a proxy in your study. All right, so if you'd like to talk more about this, like I said, I hope this left you wholly unsatisfied, but charged up to think more and talk more about this. There are some other opportunities for you to engage with this. Right now, I'm teaching advanced social epi. You can still sign up for the course, I think. It's offered this third term, and we have some modules in talking about race and ethnicity. Next month, a few colleagues and I will be, with the help and support of Katie Lesko, are releasing an SER playlist on papers on modeling race, sex, and gender. So if you're a Society of Epidemiologic Research member, you can look forward to that. Then next term, we also cover race and ethnicity in the foundations of social epi that Dr. Lattimore teaches. That's generally offered fourth term, and then on alternating years offered second term. And then next year, instead of advanced social epi, I teach methods for assessing power, privilege, and public health, where we also tackle some of this. In terms of some of the modeling as well, I think there's more work to do. You know, these courses don't just focus on race, they focus on a lot of other things. But I think it could be interesting for people across the school to consider doing a special studies to develop some more content and thinking about how the different ways in which race is modeled in studies. All right, so thank you. Feel free to reach out to me on Twitter, check out my website, and thank you again for letting me share this information with you. I look forward to the other panelists' work.
Yeah, thank you again, Dr. Dean, for the wonderful presentation on the use of race in social epidemiology. So given the limited time we have for the seminar, we will hold the Q&A session until the end of the seminar, but please feel free to put your questions in the chat box for Dr. Dean or just send her an email. So our next speaker will be Dr. Jan Wojcik. Dr. Wojcik is an assistant professor of genetic epidemiology in our department, and she currently serves as the co-instructor for the Genetic Epi 2 course. So Dr. Wojcik's research focuses on improving statistical methods for complex trait mapping and polygenic risk scores for historically underrepresented populations here in the United States. And by doing this, Dr. Wojcik aims to address existing health inequities and also to ensure downstream translation of findings from genetic epi studies for everyone. And her current work also involves the elucidation of genetic susceptibility to infectious diseases and vaccine response, and also the host pathogen coevolution. And Dr. Wojcik today will give a presentation on the use of race in genetic epidemiology research. And thank you again, Dr. Wojcik, for accepting our invitation. And now the floor is yours.
Great, thank you so much again for the invitation to speak to you today. I'm going to be speaking on how we consider race, ethnicity, and genetics and genetic epidemiology. And I wanted to start off, this is an epi department, and sort of to set the groundwork of how, just how it's currently considered in genetic work. And it is a little, what I'm going to highlight here in this brief talk is that we think of it in terms of what we think of it as a variable from a different perspective, sort of why we care about it as much, and sort of what the consequences of that has been in genomic research. So typically in genetic epi studies, these can be anything from genome-wide association studies or candidate gene studies. The analyses are typically stratified, so they're limited to one specific racial or ethnic group. It can be self-identified groups, so when you ask participants, it can be determined by genetic methods, or you can use both of them to create your groups. And if individuals are pooled, these models adjust for genetic ancestry, and typically genetic ancestry and self-identified race ethnicity. And the reason this is because genetic epi focuses primarily on the relationship between specific genetic variants and an outcome or phenotype of interest. And I'll show you in a second that the model is sort of very limited in terms of what the scope of the analyses are, and everything else is seen as something that needs to be adjusted for or controlled for, or things in your data. So it's sort of an issue with the data. Now I wanted to say first off the bat that a lot of things I'm going to show you here is how the field currently is. It's not necessarily how I think it should be or the priorities of my work, but just to give you an idea of the field and the work that's out there and being published. And so if we think of our conceptual model in terms of the genetic epi, we have the genetic variants and the phenotype or the outcome of interest, And this is what you're trying, this is the association you're trying to determine, whether it is with the GWAS, whether it's polygenic risk scores, which sum across numerous variants. And then everything else is sort of something to deal with. So the light blue strip right there is really what you're focusing on. Those are the results. That is the arrow that you're going to be reporting your results in your main text. People don't report any other terms in their regression models. It's sort of just almost like nuisance parameters. It's just there to make sure that the data isn't a result of confounding, but sort of nobody reports those estimates, for better or for worse. And you have everything else below that that sort of supports this underlying relationship, and that you're trying to tease apart the genetic variant to the phenotype. And there has been an attitude that's pretty pervasive in genetic epi, where if you can adjust out for everything else, then what you're going to be seeing is this relationship. Now, that's not necessarily true. We know this from various studies, simulations, and real, that genetic ancestry influences the measurement of those genetic variants. It influences the sort of null distribution of those variants in a population. So your genetic ancestry can be loosely correlated with the race or ethnicity that you either self-identify or people assign to you, or perceived race. And then we know there can be confounding by other risk variants. And so I want to just back up and sort of give you a little bit of background about population genetics. I'm not going to go really into it, but sort of give you the foundation of why this is. And so, as Dr. Dean mentioned, race and ethnicity in a genomic context and genetic ancestry specifically is a surrogate for historical geography. It is not necessarily a surrogate, especially in the United States, for current geography, although there are some differences, but it's historical geography. So what you see here is that, you know, over the entire genome and the millions and millions of variants that can be measured, because human beings have, you know, lived in communities that have been usually for the majority of human history relatively isolated from each other in terms of both geography and migration routes, that there's differences in frequency of variants, the allele frequency of genetic variants, and the correlations between variants, the structure of the genome, the linkage disequilibrium. And so what you can do is you can feed in sort of a random smattering of genetic variants you've measured that you know are variable across different populations. And what you get out is clustering. So here on the right is a pretty old paper at this point, looking at this in Europe, just Europe. And what you see is if you feed it into a principal components analysis and then you plot it, you get roughly the geography of Europe. And this was, people in this analysis were limited to those who were in that geographic area and also had all 4 grandparents from that geographic area. So again, historical context and historical geography. So what is this like when we look at sort of zooming out, so more of a global, you know, perspective, which is what we typically think of when we think of race. And what we see is that there are some clusterings in terms of that geography. Here you have 4 different ways of clustering people based on their genetics. I'm going to highlight the PCs on the left-hand side, which is principal components analysis. It's a dimension reduction technique where you take millions of sites and you reduce them to, here are the top 2 axes of variation. And what you see is these populations do cluster by roughly the geographical area. But I want to take it back to the difference between genetic ancestry and race, because they don't mean the same thing. They might be a very loose surrogate that could serve some purpose in some analyses. But when we think of the genetics, it's also very dependent. So for example, when we say Asia and we say an Asian person in the United States, typically that means an East Asian person. That is sort of all the way up on the top left-hand side here. But we would also group East Asians and South Asians together in one group, even though they're not really a homogeneous population. That doesn't really make any sense. If you go to the UK, the larger group is actually South Asian individuals. And so when you say Asian in a UK study, like the UK Biobank, what they actually mean is South Asian. And so from a genetic standpoint, the use of race in terms of Asian doesn't match a meaningful interpretation of the genetics when we think about what, looking at the data and figuring that out. And then also, we think of other racial groups when we think of the race of a Black participant in a Black population. Right here, you see that it's sort of loosely, on the right-hand side is the continental African populations. This is Nigeria, Kenya, Gambia. And then you have some admixed groups, African Americans in the Southwest United States, Afro-Caribbean individuals, and then you also have Puerto Rican individuals. And so that, Puerto Rican individuals over here would be classified as Black in some groups, what Dr. Dean was saying, so genetic ancestry, Hispanic in others. It sort of depends on the context what you're looking at. And just to build on this, if you look at Hispanic groups, which we'll go into in a second, they're the pink and purple colors on this. It is everywhere due to history. And it's one of the more ridiculous labels, I think, in general. But that is sort of from a genetic standpoint. This becomes even more ridiculous when we talk about genetics in terms of United States. In the United States, the non-white populations. And so what you have here is, again, a principal components analysis. This is a study of underrepresented minorities in genomics. And what you see is, again, the same axes being taken out: Africa, East Asia, the Americas, Europe, and then you have how people self-identify. And, in a sort of typical genetic study, you would want to stratify by these groups. You'd want to create these meaningful, homogeneous groups so that you can do your quality control metrics, look at your distributions, because when you clean genetic data, to find technical errors and outliers, you're comparing them to the distribution that you find in your study. There's no external sort of standard. And so you want to have a homogeneous group. However, how people self-identify doesn't really fit in those groups because these groups are admixed, meaning they have ancestry from 2 or more sort of continental groups. And so for this analysis, actually, we just pulled everyone together and used better models to deal with that. But I'm going to move on just for the sake of time and sort of get through this so we can have more of a discussion about this. But I want to even delve deeper into these labels. So what you see here is each vertical line is a person. They are colored by the proportion of their genome from that continental ancestry based on whatever references you said it was. And so here what you see is that there are groups that are on the top, how they self-identify in these large racial ethnic categories. Within each one of these groups, there is a full range of ancestries. Within the African American group, you have on average 20% European ancestry and 80% African ancestry. This is pretty standard in a lot of studies. But I want to point out that this doesn't mean that everybody has 20% and 80%. You have individuals who have almost entirely African ancestry, almost entirely European ancestry, and they self-identify as this group. And that is not an error. That is not a bug of the data. That is a feature of the data, and it needs to be incorporated as such. And then we go to Hispanic Latino. You sort of see how ridiculous this gets, where there's a lot of different ancestry. And if you go down to a more granular look at this, you see that on average, there are differences between these different groups as they identify. And that's because it's in the Caribbean versus the continental US. There's a gradient. There's a large amount of land you're covering for geography, and a large amount of variation in their history of colonization and slavery that influences their genetic makeup. But I want to point out that even though on average in these groups there are differences, if I picked a single person and I looked at their proportion of African ancestry or European ancestry or Amerindigenous ancestry, I wouldn't be able to tell where they came from, because that's not a unique feature of any specific groups. Okay, so that's sort of how we handle genetics and race in genetic research. This has consequences. So it may have come from a technical standpoint where we want to make sure that we can ensure clean data. We don't want to have errors in the data. We want to have most of the statistical methods are made for homogeneous groups. But that means that your lowest hanging fruit is going to be the easiest route, and that is going to be European populations, as it is in a lot of other epi research. And so what you have here is the mean and median sample sizes of GWAS, so these really large genome-wide association studies over the past 15 years or so. And you see there's been a rapid increase in the mean sample size for white or European ancestry groups, and really not a lot of progress on in the other groups. So these groups are being left behind in genomic research. And why this is because it's turtles all the way down. And by turtles, I mean a Eurocentric bias. There's other words for that, but I'm not gonna go here. But it's we have these large genomic health inequities that exist right now. And I'll give you 2 examples in a minute. But it's a result of the entire foundation of genomic research being built to uphold and give progress to one group and not any others. There's an idea that is just insidious in genomic research, that there's a trickle down sort of effect where if you do a lot of research in white communities, biology is all the same, right? It'll be fine, it'll trickle down to those groups, and it just hasn't proven to be true, because even though the biology may be true, your measurement of that biology is not the same for these groups because of those sort of random differences in population genetics. So this includes, when you think about from the bottom, the genotyping arrays tag European groups better. This is a more recent development where they've been expanded to other groups. These large consortia that you require for genetic studies to get adequate statistical power are predominantly European ancestry. The statistical methods that use that data are assuming homogeneous populations. This has changed in the past few years with more being built out, but it's still largely for homogeneous groups, and definitely exclude admixed groups from these analyses. And then on top of that, the functions, so how you follow up these discoveries. Functional databases are predominantly European ancestry. Biobanks for clinical follow-up for studies are predominantly European ancestry. And so you have this bias built into every single layer of this field, which results in what we have now. And what are those results? The results are just gross health inequities when it comes to genomic research. So here you have prenatal testing for cystic fibrosis and the CFTR gene, which we know predisposes people for cystic fibrosis. So when you do prenatal testing and you get the results back, there's always this little asterisk at the bottom saying this is for white, usually non-Jewish populations. And it might be less reliable for you, and that has real consequences. So here what we have is the false negative percentage of these tests by different racial groups as they self-define. And what you see is that the false negative for a bunch of these different tests is relatively low for white populations, white individuals, and it goes up and up and up for other groups, mainly due to the absence of non-white populations in studies. Now, people have said this is because cystic fibrosis is much more common in white and European ancestry groups. So what? You still need to look in other groups. Just because it's harder doesn't mean you don't have to do it. Okay. And then another example, this is clopidogrel and a CYP2C19. The CYP2C19 haplotype is associated with poor metabolism of clopidogrel. It's an antiplatelet medication. It's commonly marketed as Plavix. That's the commercial name. And this poor metabolism of clopidogrel is found in about 3 to 5% of European populations. You can see the frequency on the right-hand side. It is found at 15 to 20% of Asian populations, which means this doesn't work in those populations at a really high rate. And so this is actually the source of an ongoing lawsuit from the Hawaii Attorney General, because Plavix was marketed to their populations, which are predominantly Asian Pacific Islander, where it doesn't work. So it's false advertising. And so they're actually in the middle of the lawsuit that's been going on for many years now, but still going. So just to wrap it up, because I know we have a lot to discuss, I want to say that genetic epi usually comes at it from a very technical standpoint and a measurement standpoint. A lot of studies do not consider the downstream effects of this. They really want clean data. They want the most bang for their buck because genomic research is expensive. Every sample is expensive, and that has consequences. And so there is different meaning when it comes to race, the link between race and genetic ancestry. And I think really what we're coming to terms with, and I think people need to think about more, is that there's a disconnect between your motivations for having one group in genomic research for technical reasons and statistical reasons versus why you're doing that research in the first place, which is to have downstream consequences for public health and for precision health. And when we say precision health, we want to say precise for who, because it's not for a lot of people. So what's the point? But the idea is that moving forward, we'd have a more holistic view of this, maybe bring it more to the epi part of genetic epi, where you have both the X and the Y, your genetic variants and phenotype, but understanding it's within the context of everything else, which would bring more groups in and really move us forward. And so that's sort of where a lot of my research comes in, trying to expand who we look in, because it's not a matter of, X and Y, it's everything. And that is all I'm going to say for today. That's all I have. Thank you for listening.
All right, great. Thank you, Dr. Wojcik, for that great presentation. Again, for the sake of time, if you have questions, please hold them till the end, but you can enter them in the chat and we will keep track of them. So you can type them in now if you are have questions for Dr. Wojcik or Dr. Dean's presentations. I'm going to introduce our third and final speaker, Dr. Tanjala Purnell. Dr. Pernell is an assistant professor of cardiovascular disease and clinical epidemiology with joint appointments in the Department of Surgery, Health Policy and Management, and Health Behavior and Society. She's also the associate director of the Johns Hopkins Urban Health Institute, as well as the associate director for education and training at the Johns Hopkins Center for Health Equity. Her research focuses on identifying and addressing patient, healthcare system, and community factors that influence health and healthcare disparities for adults with cardiovascular disease risk factors. She's a wonderful mentor and teacher, a frequent and beloved panelist for our seminars for Epi Ideas, and also very involved in training public health, nursing, and medical scholars who are working to advance health equity. We're really excited to have her speak today about the use of race in clinical research and clinical risk prediction. So take it away, Dr. Purnell.
Thank you so much. It is indeed an honor to be here, I think, for my now third Epi Ideas sponsored seminar. And so I don't take it lightly that this platform is certainly an honor to be able to come and talk to you today. So I now have the last leg of this marathon talking about the use of race in clinical prediction models. And to be honest, it felt a little bit like, this was a moving target all the way up till this morning. There were still papers coming out. So similar to, the prior to dynamic speakers, you will not walk away from this talk with all of the answers, but I hope that you will walk away with motivation to do the right things for the right reasons. And so let's get started. So again, just a reminder of what is race, not to really belabor it a lot, but I think that, by the end of today's overall seminar, that's the most important thing. I think that you can walk away with both a better understanding of what, we are actually measuring and capturing, but then also, and forgive the telemarketers who decided to call right while I'm speaking, but also, just really thinking about references that you can source and cite when you may be challenged by others. And so this first slide I call Black Woman with a Headache, right? And so I'm really having to think about things like, okay, well, what do we mean when we say race? So I will tell you the latest debate in these clinical prediction models, I really did not want to touch with the 20-foot pole because there's just so much going on right now. But my mind went back to about a year ago, one of my last in-person meetings right before we shut down for the pandemic. And, very early in the morning, I think it was 8 a.m. meeting, my head was killing me, which is why you see all of these different versions of how I was feeling. And, the meeting was just going well. And one of the project managers was presenting some preliminary findings for a different study of which I'm not the PI, but she just simply said, we capture here race and gender and other social constructs. And one of the people sitting around the table immediately raised her hand and just sort of started going off and saying how she was so sick of hearing race and gender being described as a social construct. It's not social, it's biological, and that the National Academy of Medicine and the NIH published stuff that backs up her view of it. And, being the only, person sitting at the table who was a faculty member known to be, a person for which health equity is a core principle of my work, all eyes immediately went to me. So again, I'm a Black woman with a headache in this meeting early in the morning. It's too early for this. And so, I tried to very nicely point out that that is not aligned at all with the scientific evidence. And in fact, we have an entire body of research, within health equity, within sociology, within, demographics, within everything, saying exactly the opposite. And she proceeded to essentially tell me that perhaps the entire field of health equity was wrong. because it didn't fit her narrative. But in the meantime, you know, at the end of the day, the discussion ended with the PI appropriately pointing out all of the reasons why the field of health equity was right. But it also, in that moment, reminded me of why we have to know this stuff and know it so well and have so many different sources of information to back up what we're saying and it can happen anywhere, right? And so one of the things that she may not have known was that the heads of a couple of the NIH institutes were actually lecturers in our health equity research methods course. And so I just very quickly, you know, sent an email, a text and said, hey, you know, I'm in a meeting where this person is essentially saying that you all released something that race is biological. When did this happen and what's going on? And so I was immediately pointed to a few references. So the short answer is that wasn't true. But I really thought that it was interesting that you know, there were these references that have been out since like 2018, basically highlighting what happened at a working group meeting where heads of NIH institutes called together different experts in the biomedical field to say, we need to really examine how race, ethnicity, and ancestry data are being used in biomedical research so that we can come to some consensus. The short answer is there's still not an absolute consensus, because that's what I asked for. I was like, well, where's the page where NIH just says this is what race is and what is not? And, you know, this was sort of the closest that we got to that. But one thing that really stood out was this, you know, line where I highlighted in red that regardless of the fact that we need to all sort of come together across all of these different disciplines to come up with, you know, one definition. In the meantime, what we really need to do perhaps is to be mindful about how we are reporting differences by racial and ethnic groups in public health and medicine in order to make sure that we are not perpetuating the myth that race is a biological fact. And so then, you know, it made me think again about, well, in case, you know, someone decides to say, well, the NIH is wrong, let's think about what else we can sort of cite and think, you know, think more conclusively about in terms of, you know, having receipts when we have these conversations. And so for many of the studies that we conduct, you know, in clinical research, especially thinking about survey studies or looking at data from our national registries, much of what we are really measuring or have access to is based upon either self-reported race or provider-reported race, which again is just another form of, you know, sort of random identification. And so what many of the categories also are sort of following are the US Census and Office of Management and Budget Standards. And so as Lori already pointed out earlier, these categories have changed dramatically over time. And, you know, it was sort of, in a way, we're just sort of making up the rules as we go along. And it's interesting to me now that the US Census actually has a page sort of describing how they view race as it is self-identified for the census. And what they, you know, essentially says is, you know, was that self-identification of race is generally reflecting a social definition and should not be an attempt to define race biologically, anthropologically, or genetically, et cetera. Also saying that, you know, people may sort of mix and choose what categories they want to use as labels. We also know that these categories have changed dramatically over the different versions of the census, et cetera. I went on this morning and found that the US Census, you know, said, hey, we have time today. We have more receipts for you. And so, you know, I think that as a response to what's been happening in 2020 about, you know, how we need to challenge ourselves to continue to do better, you know, they've added a new page saying that, you know, we recognize that one challenge we currently face is how Americans view race and ethnicity differently than in decades past. And essentially, basically saying that they're going to keep changing this thing until they can you know, get it to where I guess the majority of society thinks that it is appropriate. But again, you know, reflecting the fact that this is sort of a moving target and how we have to really be mindful of that when we are describing differences in our studies or describing implications, right? And so you've already heard this as well too. that self-identified race or ethnicity is not a direct, correlate with genetic ancestry. I actually, I remember being invited to give a talk to a group of mostly translational scientists. And so I reached out to a friend of mine who graduated here in human genomics and said, hey, I need receipts from your field. What are you all saying? And so one of the things that she really pointed me to was several papers that sort of came out where they were essentially comparing, how does self-identified race sort of compare with ancestry and genetic data. And at least within the field of breast cancer research, they basically, reported that often what they were seeing was that there were close associations with, genetic ancestry sort of along the lines of West African descent, but not so much with East African. And here, really highlighting that there may be less shared ancestry, for example, between African Americans and Ethiopians. And so, again, just talking about how all of these things are moving targets and how we have to really be careful and be mindful and know what it is that we're measuring and what we're not when we use self-identified or self-reported markers of things like race. And so, this piece that really has been in the news a lot has been in the use of clinical prediction models, clinical equations, et cetera. So late last year, a paper came out in the New England Journal of Medicine entitled Hidden in Plain Sight, Reconsidering the Use of Race Correction in Clinical Algorithms. And so the premise here, and I encourage you to read all of these articles when you have the chance, but one of the things that really this article went in depth in doing was really pulling out several different prediction models and race-adjusted algorithms across several different fields of medicine and public health, from cardiology to kidney disease, et cetera. And it really, again, was sort of along the lines of this reckoning that society was having in terms of really wanting to better define what it is that we're measuring, what it is that we're saying when we are describing participants from a study. And so, you can do a quick search. There's lots of papers out now. For example, Misinterpreting Race, the Role of Medical Schools in Propagating Physician Bias. There's another one on Race and Genetic Ancestry in Medicine, A Time for Reckoning with Racism. And then, even recently, Racism, Not Race, Drives Inequity Across the COVID-19 Continuum. So lots of discussions going on. As many of you know, my field is most closely associated with kidney disease. And so even prior to 2020, there was a paper that came out in JAMA in 2019. And, it really, the title was Reconsidering the Consequences of Using Race to Estimate Kidney Function. And so this started, a lot of debates and conversations in the field about essentially, as you may know, we use estimated GFR for lots of decision-making, for example, when to refer people to see a nephrologist, whether or not to classify someone as having chronic kidney disease. These, are things that may have implications, for example, in terms of who could be considered living kidney donors or who should be on certain types of medications that, need to be processed through the kidneys. And so lots of conversations sort of started prior to, spring 2020, but certainly have continued. And so, again, some of the papers that have come out recently, removal of race from the estimates of kidney function. And so not only, debates about whether it's the right thing or the wrong thing to do to remove it, but, really the implications of should we replace it with something else? If so, what should that be? Recently, I was asked to participate in an editorial where we talked about sort of some findings from a paper that basically looked at using a different surrogate, really swapping out race and looking at different measures of creatinine, for example, and trying to estimate kidney equation and sort of the implications of that. And so, really a lot of this is sort of rooted in the fact that, our two, most widely reported equations, as you may know, are the MDRD and the CKD-EPI equations. And so those are, two widely used equations in clinical practice. And sort of part of what is driving the most recent conversation is that I think several medical students and others started pointing out the fact that when you get labs and routine labs, you'll see two different numbers. One that say, if you're Black, this is your number. If you're otherwise non-Black, this is your number. And so lots of conversations about what that means and what it should mean and what it should do. So, just in the last couple of minutes, I'll talk about the fact that, as I told you, I didn't want to touch this whole debate with the 20-foot pole, but you will learn that as you continue to occupy spaces and your work, gets to be known, and, you have a voice and you have to use it. You have an obligation to use your voice. So one paper that we published in JAMA in 2018 basically, described the fact that we have worsening disparities in access to living donor kidney transplant using some of the national registry data. And an editorial accompanied this and basically posed the question, is it really race, or is it poverty, or is it, income or other surrogates? And, we absolutely agree that it's all of these things that are being measured, plus many other reasons why we see this. And, one of the things that I often highlighted in my talks was that, even years before that paper, we had published other papers looking at potential mediators and what was sort of explaining the variation in access to living donor kidney transplantation. And what we found was that, measures of, pre-dialysis care and measures of neighborhood level income and health insurance played a much greater role in explaining these disparities than did any type of differences in biology or even chronic medical conditions. We also have had a number of people for years talking about we need to do a better job of describing the role of racism and sort of propagating many of these disparities. And, in particular, you hear a lot now about structural racism and what this means and what we need to do to tackle, once we report these differences, how do we fix it, essentially? We talked, recently about the nexus of kidney disease and COVID-19 and structural racism, and how all of these end up essentially causing many of our patients to sort of have what's called like this weathering hypothesis that really talks about, the cumulative effect of all of these different barriers over time and what that sort of does. But then, finally, I'll just tell you, we are still, dealing with this in very real ways. Many of us are now parts of different professional societies or groups, but there are even ways for you to get involved. Like many of these task force that have been basically asked by Congress to make recommendations about what to do are now having public seminars, public forums. So, if you feel that you have an informed opinion about this, I encourage you to read more about, each of the different fields and ways to get involved. But also, I encourage you to learn more about the what to do. It's not enough for us to just simply describe differences by any subjective measure. We have to really be committed to doing something about it. And so I hope that that is, also a key takeaway for you, that, we have a responsibility to do something. And so that ends my talk, and I thank you for listening.